Hello, thanks for visiting my poster. So my project studies the fate of oligodendrocyte precursor cells, or OLPs, in the adult mouse brain. OLPs represent 5% of all cells within the brain. And during adulthood, they are constantly dividing and producing cells, and in particular, myelinating oligodendrocytes in response to learning or pathology. But what is less clear and hotly debated is whether OLPs can give rise to other cell types in the adult brain, including new neurons. And so to understand the fates of OLPs in the adult brain, we used the fate mapping strategy with double transgenic animals. These animals were PDGF receptor alpha, CRE-ERT2, tau membrane GFP, or ROSA26 YFP mice, where GFP expression is under the OLP-specific promoter activity, PDGF receptor alpha. So this double transgenic animals allowed us to briefly induce expression of GFP in OLPs, and then follow them up over time to see what type of cells they produce during adulthood. So during a routine examination, we were surprised to find a population of GFP-labeled neurons in the lateral entorhinal cortex, or LEC, of mice. And the LEC forms part of the hippocampal circuit, and it's important in object-related memory. These LEC neurons also show morphology of typical stellate cells. We reasoned that if these neurons were constantly being derived from a precursor population, we should expect that they would accumulate over time. And this is exactly what we saw. We observe a gradual accumulation of labeled neurons over time, indicating that they are continuously being produced during adulthood. Moreover, these newborn neurons also projected to a known downstream area of the LEC, the hippocampal dentate gyrus, indicating that they formed part of the hippocampal circuit. And finally, we also found that these neurons expressed double cordon, a marker of neuronal immaturity, suggesting that these neurons may be newborn and immature. So in summary, we've identified a group of immature LEC neurons that are putatively derived from a precursor population expressing the OLP marker. We're really excited by these findings as it suggests that there's a continual production of LEC neurons in the adult brain. If you would like to find out more about other experiments that we have done, such as characterizing the molecular properties of these neurons, quantifying their accumulation over time, or carrying out gene deletion experiments to disrupt myelination, please come by and check out our poster. Thanks so much for listening.